It's a wonderful talk. So our next speaker is Dr. Shelby Ellison, and she's joining us from University of Wisconsin-Madison. Dr. Ellison's research program combines field and greenhouse studies with genomic and genetic transformation tools to accelerate breeding progress in hemp. Her work is dedicated to characterizing genetic diversity and improving seed production systems to support the sustainability of emerging crop markets. With a background spanning UC Davis and National Science Foundation, she brings a wealth of genomic expertise to the field of alternative crops. Please join me in welcoming Dr. Shelby Ellison.
I'm gonna get my talk. Okay. All right. Hi, everyone. Thank you so much for the invitation to be here. I am enjoying this balmy weather after being in Wisconsin all winter, so this is great. So thanks to the organizers, thanks to Corteva for putting this on. You're going to hear about hemp for the third time today, so I hope you're ready to keep hearing about hemp. Okay, we've got a slide advancer here. Okay, yeah, so I'm talking about hemp. I started my research program in 2020. So really what I'm going to be doing is talking about what it's like to breed or to start a hemp research program as a new faculty, hopefully to be tenured soon, and kind of what I've done to start this program in the public sector. So I also wanted to mention, this is not my first time in Texas A&M. So I actually came to the NAPV meeting back in 2011 as a grad student working in bean. And I was very excited because there was a place called the Dry Bean Saloon. So that's great. But you hear my background a little bit. I'm from Wisconsin originally. I went to UC Davis for graduate school, working in common bean growth habit. Then I came back to Wisconsin. I've worked in carotenoid genetics and genomics. I've been involved in several genome assembly projects, and now I'm at Wisconsin again, working in alternative crops. But you're going to hear more about beans too, because I'm going to bring them in as an example, because I just can't get away from working in beans. So first, I'm just going to talk just one slide, again, about what the difference is between cannabis and hemp, or if there is a difference, and then talk about how I've initiated my program, including starting from scratch, so where I obtained germplasm, and then the characterization efforts that we've had in my lab so far, looking at specific examples of cannabinoid accumulation and disease problem, Fusarium, and then where we're going next. So, all right, what is cannabis sativa? This is our one slide. You heard this from Larry earlier. The one thing that I've added on there that we talked about is that it is dioecious. So there are male and female plants. It's wind pollinated. It's photoperiod sensitive. So in Wisconsin, a lot of our lines, we're going to have long enough nighttime lengths coming in late August, typically, where we start to see flowering. So it's grown for multiple uses. So we kind of have independent research projects focusing on the different market types. So growing for cannabinoid content, that's coming from the unpollinated female flowers. That's where you're getting your THC, CBD, CBG. The legal definition here of what hemp is as defined in the Farm Bill is that hemp has less than 0.3% total THC. So that's what we work on in Wisconsin. That's what you all work on in Texas, where we just have a federal hemp program. But of course, as we heard earlier, it can be grown for also its grain or its fiber, which are the other traditional uses of hemp. So a multitude of things that we can do with this plant. So it's an exciting emerging crop to be working in. So as you begin your research program, in 2020, what a great time to start. I did not take over someone's hemp breeding program, right? There was no hemp breeding program. So to start working in this, we need germplasm. There wasn't really any sort of cannabis industry in the Midwest. On the coast, there were medicinal and recreational companies that have popped up. That wasn't really true in Wisconsin. So the first step, of course, to start the program was to acquire germplasm. And why this is very important to me is because I'm from Wisconsin originally, and growing up, everywhere you would go, you would see ditchweed, which is feral cannabis. So the history of this is we used to grow a lot of hemp in the United States. And indeed, the last big spike in production occurred in the mid-40s here, and that was tied to World War II. So during that period of time, we needed a source of domestic fiber production. So we had this government-sanctioned program to try to grow a lot of domestic hemp for fiber. And so you see this production peaked. A lot of that occurred throughout the Midwest in the United States. And so a lot of that hemp escaped from production and became fertilized. So feral crops are just plants that have escaped from production and then are growing in their local environments. And so here it's kind of hard to see, but you can just see what a typical stand of feral cannabis might look like. So I was like, okay, well, that seems like an interesting resource that I should tap into. So was able to get funding for this, and the idea was to collect this seed to kind of kickstart our domestic source of germplasm. Because yes, there was, keep in mind this is 2020, so there wasn't even a hemp germplasm repository at this time. There were no seeds. The seeds that you could get had a lot of IP tied to them, so they were owned by private companies and people that had risked a lot to get that germplasm because it has been illegal previously. So we got some funding to make collections of these feral materials across as far as a geographically distributed region as we could. So how does one find feral hemp? It's not like you can talk to people and they might tell you if they have it growing in their back 40, but, oh, I forgot to say howdy. I was going to say howdy. Okay. I was supposed to do that. Okay, we got it by slide 7. So how do we find these feral populations? Okay, how many of you have heard of iNaturalist? Some of you. So iNaturalist is a app that you can show different where you observe biodiversity. So people go in, they look, they find different plants, animals, they're looking at insects, everything, and you can go in and take a picture of it and say, okay, I just saw this in this location. And so I was like, oh, I wonder how many of these exist for cannabis on iNaturalist. And what you see here is quite a few observations have been made. And this is from, I think, 2020, this slide. So all of those red dots indicate where people have made an observation of cannabis sativa. And then if you click on a single link, you will actually normally be able to see an image of that. You'll get the geo-coordinates, as well as who made that observation. And if you're pretty active on iNaturalist, it's actually quite a community. Lots of people like to share information, so you can actually message these people. And so we use these as kind of our starting points of places to explore, to look. And then I also painfully had my postdoc on the project kind of go through these and figure out what was actually likely feral and what might have been a cultivated field or what might be in, downtown Manhattan, which is probably not feral hemp. So we went through and pretty much characterized the ones that we thought were likely to be feral. And then we reached out to a lot of those collectors to verify if those populations still existed or if they would be willing to help with the project for the collection. And that was great. We found a lot of people were really interested. So after identifying these sites and identifying collaborators, which I think was very important in the grant, we actually said that we would pay people to make the collections and to ship us seeds, so they were motivated. But a lot of people on iNaturalist are just very happy to be involved with biodiversity conservation and collection efforts, so that was great. And I'm talking about this, we're talking a lot about phenomic and genomic data, but I also would like you to think about things outside the box of things like iNaturalist and other data that exist that you can tap into and other human networks that you can tap into to kind of get into this more multidimensional aspect of our jobs. But anyways, we developed a protocol of how we wanted this collected. And the idea was to make it as simple as possible. So the idea was for every individual female plant, you'd want to collect these at the end of the growing season. The goal is to take a 10 centimeter piece off the top inflorescence where you would be able to get seeds that were there. You were able to get leaf tissue that you could then genotype, as well as that is the inflorescence that you would use later for being able to detect the cannabinoid profile. So it was all there in one little brown paper lunch sack you could send off for processing. So the results of that were quite successful. We had, over 50 different people that helped make these collections across 12 different states. Obviously, we were highly sampled in Wisconsin because that's where we can drive to a lot to make a lot of the collections. But we did get very broad distribution across most of the Midwest where we think a lot of these populations existed. So we had over 1,800 individual samples from over 100 unique populations we collected. So that was exciting because we went from having nothing to having quite a bit of stuff. And the idea behind this is maybe these fertilized populations also have become adapted to the Midwest, where they have some interesting allelic variation that might help them in breeding programs for, regional stresses that we might have. So be it abiotic or biotic stresses. So we now have this data set and we can start doing some fun stuff with it. One thing that we were really interested in is kind of seeing where these feral populations sit within the global collection of cannabis. So since 2020, we now do have the seed bank in Geneva, and there are more accessions that have been contributed. It still is very difficult to get some of that germplasm out of the seed bank because some of it is not compliant. There's also limited numbers. Some of it is still under IP. But there is more available now. And so we were interested, this is just using two other large data sets that have a good number of European and Asian representation of different cannabis types to see where these feral populations sit in. And lo and behold, what we were expecting is these are escaped fiber types, and that is what we see when we look in our principal component analysis is these blue, green, brown, purple. These samples are all feral cannabis. So this lime green here, that's going to be the hemp fiber types, and typically what you'll see is the Italian fiber types that were the ones that were first brought over to start the US breeding programs in the early 20th century are what are closest to the feral types, as you might expect, because that is where they escape from production. So that was exciting to see that and fit that into the broader scheme of things. But it's also exciting because these are unique populations still. There's population structure. They have different unique alleles, so maybe we can utilize those in our breeding programs here. So we've got some seeds. They're not all exactly the same, so that's great. All right, so what do we do next? Well, I don't really know what's going on in hemp in 2020, so we have to kind of figure out what are the major priorities for growers, what are the traits of interest, what should we do next? So I've been involved with a couple of national surveys asking growers what their biggest concerns are. It's the same things that are the big concerns in most crops, with the exception of compliancy. Most other crops don't have to deal with that. So to grow hemp, if you exceed that 0.3% THC threshold, you typically have to destroy your crop, which is not great because you just put a bunch of time and energy into something and now you have nothing. So compliancy is a big concern. There's a couple of publications, including a collaboration here with Dr. Baltensberger, where we've surveyed growers about these things. So we have some idea of what growers want us to work on. So the first thing I'm going to focus on is, okay, let's think about this compliancy. So just very briefly, what controls THC content in cannabis? So if you look on the left there, that's like a very stylized version of the cannabinoid biosynthetic pathway. So that CBG molecule, that is the precursor of all the other cannabinoids. And typically, so there are these two different enzymes. So you have your THCA synthase, your CBDA synthase, and if they're functional, they're going to make that product. So cannabis is typically thought about as types, type 1, type 2, type 3, type 4, type 5. If you have these functional THCA synthase, 2 copies of this functional synthase, you're going to produce a bunch of THC. If you have 2 copies of the CBDA synthase, you're going to produce CBDA. And if you're heterozygous or if you have 2 functional copies of both of them, you're going to be intermediate. And that is because when you map the cannabinoid synthase genes, they actually are very tightly linked on chromosome 7 such that there's not recombination that occurs within them. And so they're acting almost as a single genetic unit of inheritance, but rather they're just linked tightly together. So what happens is these type 1s, they're linked, they're in repulsion with a functional copy of this. So if you have 2 copies, you're going to have high THC, low CBD, and if you have 2 copies, you're going to have high CBD, low THC. So ideally, that's what you want in a hemp production system, so you're not going to make a bunch of THC. Whether you get that or not, the good thing is we have all of our seeds, so we're very interested in seeing if they had these active THCA or CBDA synthases. So we extracted DNA from all of those feral samples that we collected. So Larry's lab so kindly made these really nice PACE markers that we're able to genotype for those functional or non-functional CBDA synthase. And so it'll just cluster when you run the PACE assay to determine whether it's going to be a type 1, type 2, or type 3 plant. So we ran that across all of our feral material. And what we found here, so if you look at the populations, if you see more black in the pie chart, that means that it has this kind of non-functional CBDAS allele, which means it's a THC type. If you have white, it means you have a functional CBDA. So you want to see more white in those pie charts, meaning that those are more likely to be compliant. So if they're homozygous for that, then it's going to be the type 3 plant, which means that it's ready to go for breeding. It's less likely that it's going to produce excessive amounts of THC. So of those feral materials, approximately 80% are that type 3, which is great. It means that they can immediately be used. You don't have to do any cleaning up to start working with them. And yeah, also these populations here, the way that it works right now, so different labs do have, so the USDA germplasm repository can accept non-compliant material. So there's ways that they can clean up that material then to have it become type 3, so other breeding programs can utilize it. So that was good. And this makes sense, right? Because these were fiber types. So we would expect that they actually were type 3 and would have low THC. Although, back in 1940, they were not genotyping their plants. So that's why you see some of that low level allele of the THC type. So compliancy, we're pretty happy with that because we have this germplasm now. We know most of it's compliant for breeders. So the other thing that I'm going to focus on for the rest of the talk is disease. I'm not going to be talking about powdery mildew. Larry has that covered. So we wanted to think about, okay, we know that when I first started, everyone would be like, hemp doesn't need anything. You don't need to water it. It doesn't have any diseases. That's a lie. There are lots of diseases in hemp, what we've observed. So a survey done from University of Kentucky in Nicole Gautier's lab. So she looked at different regions of the United States and took a survey of what were the prominent diseases. And all I'm going to point out here is, so this is percent occurrence. So these things are happening very consistently across these regions. And one thing that we see here that's popping up a lot is this fusarium bud mold or head blight. This is a problem in other species as well, grains, wheat and barley. And we see that this seems to be a problem. And we also, in our lab, we've had lots of samples we sent to our disease diagnostic lab. It always pops up as being fusarium. You also see that botrytis bud rot in blue here. This pops up a lot. And actually what we're observing is a lot of the times people are characterizing things as botrytis that are actually fusarium. So I think these two diseases, when you're phenotyping them, can look quite similar and people are confounding which they are. But either way, we know that fusarium head blight seems to be a problem. So during some of our cultivar trials, we see this out in the field. So Dr. Derek Grunwald is a postdoc in my lab who is a plant pathologist by training and has really brought all this fusarium work to my lab as a geneticist. And so he isolated samples from the field and grew them up and then, performed Cook's postulates, which I think is a fun thing that it's like something they tell you in school, but it's like really what you do in real life too. It's great. So you take this, isolate it, you can sequence this, and so we knew it was Fusarium, but we were able to characterize this as Fusarium gramineae as the causal agent for the head blight that we were observing in hemp. So you can then reinfect and see the symptoms and then re-isolate. So we're able to confirm that that seems to be the cause of the Fusarium that we're seeing so much in the field. So we have a more defined target from what we need to breed for or do more analyses in. This also is very problematic just because Fusarium gramineae can produce mycotoxins. So right now we're at this kind of stage in hemp production where we're trying to get people really excited about eating hemp maybe or feeding it to animals. So we definitely don't want to have like a negative PR thing come forward about, oh, there's Fusarium and there's mycotoxins and everything's getting sick. So, this produces on the plant. We make either food or feed for this, and then humans and animals can get quite sick if they ingest a lot of these mycotoxins. So there's a definite need to find resistance or integrated pest management to deal with Fusarium in hemp. So now I'm going to get into the phenotyping portion of the talk. So we have germplasm, we have some ideas of what we want to look at to research in the program. So let's start it. And I'm going to kind of talk about this in the terms of the multidimensionality of the theme of the symposium. So when you think about phenotyping for disease, as we heard today, we kind of have these traditional tools that we've done, right? You have something in the field, you have some sort of 0 to 9 to 10 to 5 scale, or you can have like a percent of the grain head that is infected that you're characterizing, or the disease progression. So this is kind of the standard way of phenotyping, and we do this in the field. We have our mapping populations out there, we have association panels, all of it. We give them numbers, we give them ratings over time. And again, from the Gautier lab, they have developed a protocol that we follow, so we're doing it the same way across different hemp labs. And so that's how we're doing our rating. But we now know, right, we have additional tools. So we've talked more about, okay, I am not a plant pathologist. So when I'm out there in the field, I don't know what I'm doing when I'm rating. I'm like, oh, it looks bad. Just write the word bad. Put that in the GWAS. So we want to move away from that. Derek is obviously way better as a pathologist. But we talked also about kind of these higher throughput methodologies. So we're calling this 2D. But this kind of takes away the subjectivity of it, right? So there are many, many protocols established where you can binarize the image or do something with color processing where you put your infected materials down. A Fusarium, like the inflorescence is difficult to phenotype, but also we actually observe Fusarium on the grain. So this is something that we're doing in the lab right now. We have one of these Vibe seed analyzers, and they already have protocols established in wheat. So we're trying to extrapolate that into hemp with the idea that you get some quantitative measure of infection on the seed that you're measuring. So great, but that's not what I'm here to talk about. So we're moving into what I'm now calling 3D, and this is going to be the rest of the talk that I'm going to focus on, and that is the world of metagenomics. So my background is in genetics. I like to think about DNA and sequence, and so I think it was during COVID, you're talking about, how people were testing wastewater to look for COVID, and how I was like, what other stuff do I have that I can parse through and look through to see if I can find something interesting, pathogens in plants. So we have several microbiome projects where we're looking to see what is the community of organisms in a specific environment, and that's very similar. So if you've thought about microbiome experiments, that's essentially what a metagenomic experiment is, but you have some community of organisms, and the idea is that you're not selecting one of them, you're trying to see what is all there. So you can take that sample, extract DNA, and then through essentially mapping to these very, very large reference data sets, you can characterize what is the abundance of different species that are present in that whole sample. So the idea behind this, and this is where it gets really bizarre from here on out, so I'll be curious to hear what you all think about this after I present it, is so normally when you're doing this, you're targeting something. You're doing a soil microbiome sample, you're targeting samples, or you're targeting the actual microbes. this is not what I am proposing. I am actually proposing I have thousands and thousands and thousands of samples that I have sequenced, and I know that typically when I get those sequences back, I throw about 20% of the reads away that are not mapping to my reference genome. And I'm like, what is that? So I'm like, more than likely, it's whatever has interacted with the surface of that leaf. It could be microbes, it could be me, it could be a number of things. It could be, insects walking around. So the idea is, what is in that leftover stuff that I'm throwing away from every single sequencing run that I do? And could we use that potentially as a proxy for a pathogen phenotype? So let's see. So now I'm not going to talk about hemp anymore. I'm going to talk about beans because I also work in beans. So I have a graduate student who's working in root rot, specifically caused by Aphanomyces eutyches. And so we have a lot of resources going into phenotyping root rot in common bean and a really, really nice established protocol. But I thought maybe this would be an interesting sample set for us to see how well this actually works. And we were far enough along with this project. So what Rebecca did is she had field samples where we have a real population, and that's just natural inoculum. So we're not actually inoculating those beans. We have a root rot field that we grow our bean trials in, and the idea was to sample the leaves, roots, and the hypocotyl for DNA extractions, and for her to just do her standard rating of 1 to 9 for disease infection. Hypocotyl is important because the Aphanomyces is attacking normally the cotyledon of the plant. That's where we phenotype typically. We might look for oospores as well, but the cotyledon is what we're scoring. And then in the lab where we have our standard protocol, that's where we actually inoculate with Aphanomyces and did the same thing. In this case, we sampled leaves and hypocotyl and scored those materials. And the idea was to see what's in there besides bean DNA. So we already do this, right? We have our phenotypic score. We extract DNA from our bean samples. We run our QTL mapping. That's great. That's worked. We have QTL. We know what's going on with that. But what else is going on with those thrown away reads? So I work with Elliot Stanton from my lab, who is a data scientist. And essentially, he comes from a microbiome background. And so the way that you do this is you initially throw away all of your reference genome. So in this case, you get rid of all of the common bean reads. And then with the remaining reads, you're going to map those to a database. So we use something called Kraken, which does the taxon classification. So it takes those reads, tries to match them. You can change all kinds of features to see what level is accurate, or classifying them at the genus or species or family level. And then, the idea is to look at the relative abundance between all of your samples with those classifications. So just getting into a little bit, and this is all quite preliminary. This is things that we're trying right now in the lab, and the good thing is, it doesn't cost anything. It's just our junk DNA, so we're not really missing anything. But the first thing that you can see is if you look across the different tissue types, so our root, leaf, and hypocotyl tissue, so here's a measure of alpha diversity. So you can immediately see that we're seeing much less diversity that's happening in the above ground tissue than the below ground tissue, which is typical of what you see. There's a lot more going on below ground. So that's captured here. And then the rating here is just if something is resistant, susceptible, or intermediate. That doesn't matter in this case. But we definitely see that our leaf tissue has less going on than our root tissue. And this goes back, so, the short read archive is filled with sequence that's just sitting there. And I can't even begin to imagine different things to test, but I do think that there's something to, we're troubleshooting this idea of normally we take healthy young leaf tissue. That's what you write in every manuscript, right? Healthy young leaf tissue was extracted, or you extract DNA from it. In this case, you want to take diseased, icky tissue at the end of the growing season, because you want to have all of those pathogens present. So kind of starting to think, this maybe is a little bit more like RNA sequencing than DNA sequencing, because maybe you want to get at the tissue type or the where you're seeing the pathogen. So this is something for the future, but we are seeing differences here. So getting into the initial analysis, so I'm going to explain what's going on here. So we've got a heat map, we've got some hierarchical clustering. So what I did is from those samples, we extracted all of the species from the Oomyces because it classifies them into species. So there's like 4,500 rows of data of species classification that you have read counts, right? That's how many reads are getting mapped to that particular species. So you can classify it, and then you can take a relative abundance because you can look at the total number of reads for each individual sample. That's going to be important. When you get your data back, you know each sample is not getting the same amount of sequencing. So you can normalize it by read abundance. So one thing just to see here is, okay, we had our field and our greenhouse experiments. So what we observe right away here is that this is Aphanomyces euteches. That is the genus species of what we inoculated those plants with. And you see the susceptible roots grown in the lab have the highest relative read abundance of that species, which is great. That's what we're interested in. So something interesting is happening here. Another thing you can see is all these other Aphanomyces species are popping up and things from the same family. And this is where you get into some of those like problems with detection. Maybe we're not able to classify it as a species. Maybe we have to go to the genus because these, what the difference is for these short reads, they're messy reads, there are not many of them. So we do see that it's not perfect at identifying the correct species because whether these things were actually there, probably not. It's probably just read misclassification. So thinking about that is one thing. But overall, we're very pleased that we see the most things that are being identified as Aphanomyces euteches. So that was great, super exciting as a proof of some concept for, I don't know. The other thing that's really interesting is then when we look in our field samples, and I think this is where this becomes more multidimensional, is, okay, we're in the field and we have this cluster of susceptible samples and they do have AF. We do see small amounts of AF in those reads, but we also see Pythium. So this is very interesting because you're going out into the field and you're doing your 1 through 9 score. Well, what are you phenotyping? It's not like you're there with a pathologist and you're running diagnostics and verifying that what you're actually phenotyping is what you think you're phenotyping. You're just phenotyping that hypocotyl on this 1 through 9 scale. So in this case, it looks like in these field-grown samples where we know there's AF, but we are still picking up something else. So we're actually phenotyping a whole nother organism. Through these reads, we're able to show, I don't know if it's AF or it's this combination of AF and Pythium that we're actually phenotyping. So it really gets at this idea of sometimes it's more complex than just one thing. And indeed, when we look at the root rot complex in common bean, Aphanomyces is the predominant root rot pathogen in Wisconsin, but it's frequently found in complexes with Pythium and Fusarium solani. So this is exactly like, we know this, but this is a way that we're actually picking this up. So starting to think about this phenotype, not only as a single individual, but maybe you need more complex layers to the phenotyping or the interaction of these two pathogens that is causing the final phenotype. So just to show you that, it's super interesting. We're really happy to see that. I don't know what we're going to do with it yet. Talk to me about it because this, like I said, this is new stuff we're doing all in the past couple of months. Other things to show from the data, just as we're starting to probe this, we have this on hundreds and hundreds of samples, is, okay, so here, this is again, relative abundance. This is now by order. So remember our kingdom, phylum, king, pack, could, order, family, guy, special. I remember how I used to do it in school, but we have leaf tissue and our root tissue, okay? And then we don't have lab root tissue that we're looking at, but just lab and field leaf and root. And so if we look at these order here, one thing that's very interesting that pops up right away is this is Phaseolus vulgaris. So we're seeing a huge abundance of the reads across all these samples. Even in the leaf, we're picking up rhizobium. So we're seeing, those symbiotic those very common symbiotic microbes that we're finding, definitely prevalent in the root, but very interesting even in the leaves. Other things that are interesting, so the leaf lab samples, a lot of stuff we're picking up there, including this species, which this is like a human communal pathogen, or not pathogen, it's a microbe very commonly found on human skin. So definitely you're gonna start picking up things like that are gonna be contaminants. In the lab, more likely we're handling those all the time, we're inoculating them. It looks like this is very, very diverse, but you have to think about this also. The leaf tissue, most of the reeds in the leaf tissue actually turned out to be Phaseolus vulgaris. So there's fewer reeds that go into the junk pile compared to the root and hypocaudal tissue. So you also have a lower number of reeds. So there's also, there might be 1 million reeds in here and 10,000 reeds in here. So that also changes the proportion of things. And this is things that we're grappling with. But ultimately what we're seeing here is that we are starting to see there is microbial composition that differs both by tissue type, but also by environment, and how that's important and what that means and how we're going to start to model these various factors as we think about this. But it is somewhat promising to look at a bunch of different samples and see that they have similar composition. And we're going to start doing this in higher repetition to see within a single sample how much variation you might be able to see, and how things like read depth, when we're sequencing factors, these composition factors into the composition. And then, I don't know, what time is it? Does anyone know? Okay, great. So one other thing, just going back to Fusarium again, because I just took this little path into beans and root rot. So going back to these Wisconsin feral samples that I had. Okay, so these were collected. They came in those little 10 centimeter bags, and then we genotyped them. And so I was like, okay, well, let's see what's in those samples. So if you look at this, so these are individual samples. This is now separated by different genus. So I think this is the top, I don't know if it's the top 30 most abundant genera that we observed in these data sets. And so if it's red, it's in a higher abundance. If it's blue, it's in low abundance. And so what you see, going back to our disease survey of what's very common that we see in the Midwest, everything that's circled there, Alternaria, Bipolaris, Scospora, Fusarium, all of these things we're able to pick up. So the most prevalent diseases that you would find in cannabis, we're able to pick those up. And you can also see some of them are in high abundance, some of them are in low abundance. But then you can get into interesting things like, okay, if you don't see it in a population, what does that mean? Is it not there? Or are those samples more resistant to it? So we're trying to think about what we're going to do with this. But we are happy to see that when we look at that junk DNA, we pick up what we assume we should be picking up. So that's been promising. This is also, again, going back to that time and place, these are dead flowers that we're collecting. We want the seeds to be mature on them. So we're not picking up diseases that you might see mid-season or something, right? Those are going to be long gone by the time I'm collecting these in October, and you have mostly dead tissue, so a lot of more necrotroph type organisms are going to be found there. But nonetheless, very promising to see a lot of those high priority diseases. There are reeds sitting around in those, in that junk. So, yeah, this is all new. We just did this. We're doing this in a much more involved way now. You can parse data sets online and kind of figure out what are optimized data sets for this, if there's disease rating scales, if they took it from, so a lot of the RNA-seq data is very useful because we actually know whether those materials have been inoculated and start to see, can we actually use this as a proxy for a phenotype? So we're doing this right now. We have field trials. We have greenhouse trials that we're inoculating with Fusarium, where we're collecting fresh tissue, we're collecting infected tissue. We have the phenotyping, we have the imaging. So we're trying to put this all together to see how well they all correlate with each other. It's not really that, if it doesn't work, it doesn't work. We have the phenotype, so it's just trying to do something with things that exist that we're not doing anything with currently. And then, yeah, looking at the correlation and the heritability of these traits, the ultimate power with this is that we're no longer phenotyping one thing at a time. We're phenotyping thousands of things at a time when we do this, because we have reads from thousands of different organisms, and really getting at these interactions of different species that are found, thinking about how we can better model that in our phenotype when we're trying to make selections and trying to get a higher estimate of heritability for a trait, which maybe isn't caused by a single factor. So there are huge limitations to this, okay? I'm not gonna put all my eggs in this basket. Clearly, there is a lot of contamination in these samples that you might make an assumption from that it's not true. So, we're finding a lot of but the thing is, you can filter all of that out. You can pick any genus or anything to focus on and see what is there. You obviously need to normalize this, like I said, by the number of reads in each sample. As we mentioned, DNA genotyping, the sequence cost is going down. It's the phenotyping that costs a lot of money. So we can increase the number of reads we want per sample by 2 for not that much more money, to try to increase actually the coverage for these other organisms so we can get a more accurate representation of what is going on in this metagenomic data. So it might be difficult if things are there in low abundance, we might not be able to capture them. And the annotation issue, you saw, so we're able to define things as Aphanomyces eutyches, but we're also getting some things called other types of Aphanomyces. We're seeing that time and time again, that it's very difficult at the species level to get classification. You might have to go up to the genus or family. But again, that might get better as we have better annotations for genomes. More genomes go into the Kraken database, or if we use longer reads. Right now, this is all being done with short read, Illumina reads, so we'll see what happens as we move into longer read technology. So you might have not known, but there's been 2 steps discussed already. So we had germplasm, we had grower concerns that we're working on. That was the cannabinoids, the fusarium. And then what I like to so cleverly call infinity for our multidimensional thing is, the germplasm is, to me, the multidimensional factor of this. Everybody talked about all the collaborations they have and all the people that they work with. So I'm very happy these resources, so they now sit in GRIN, and so everybody has access to that material. It's been used in a large number of programs, so there's breeding programs that are utilizing this. We have a partnership where we're initiating breeding programs. A lot of these materials have been used in pangenome efforts and continuing to be phenotyped in GRIN, and all of the data will be publicly available. We also work very closely with the Crop Innovation Center and are validating many of our traits of interest through that amazing facility. We utilize this material. We talked about GEBVs, so that you can also look at genomic estimated adapted values. So of these populations, are there interesting alleles that seem to be important for different climatic features? So we're parsing that, looking at species distribution and modeling where feral hemp exists currently, and then under different climatic, increased temperature, what's going to happen to the distribution, where should we collect from, where should we look to move into the future? So really the idea is that this material can be used broadly by the hemp research community, including any of you that are interested, and that's ultimately how we're really scaling up the dimensionality of the program. So, concluding remarks here. They're generic, they've been said already, but ultimately building intentionally collaborative teams. So I think it's really important. No single person can do all of these things. Like, you hear these talks, and I feel as a grad student, it's quite intimidating. You're like, how can one person do this stuff? And they don't. I think that's like the thing I've learned over time is, you just work with amazing people that do things that you can't do. And if you do that intentionally and well, then you can do really amazing stuff. I have co-create research that matters. I always think that our research should be really centered in what growers need and want. So never losing sight of kind of what is most important to them, because I work in emerging crops. I don't know what they want. They have to tell me what they need for me to work on it. So I have this, we're talking all about innovation. and data science. And I think one thing that we're saying here is remember that some things work well and that we don't need to innovate in those areas. It goes back to thinking about what humans do well and what computers do well. So just to make sure that if things work really well in your program, that you should keep on using them. And ground truth everything. Again, you can have these kind of like harebrained ideas, but just don't build your entire program on them, that they're truth and that they're working. Make sure that you ground truth everything. And yeah, with that, here's my lovely research team up in Wisconsin and all the collaborators that I've mentioned, various parts of their research in the talk and different funding agencies. And I'm happy to take any questions.
So really interesting. I'm curious, have you done any correlations with, just your susceptibility ratings and the actual pathogen that you're looking for, or more broadly with just overall, OTU abundance or diversity with some of the other traits that you look at, maybe like vigor, if there's more microbial diversity, the plant does better, that sort of thing?
Yeah, so the only thing that we've done so far is that we've looked at correlations between our numeric scale and our read counts, and they're very highly correlated. So we have like a correlation of like 0.85 on like read count and that 1 through 9 rating. But we haven't looked at anything as a community or network. So like when you do that so that's the lab samples. With the field samples, it starts to fall apart.
If anyone has a question, please come up to the mic and queue up with the mic.
That was a great talk. So my question is, have you tried to isolate these pathogens from the samples before sequencing?
Have we tried to? So, other than we frequently will go to the field and we'll take samples and bring them back to the lab. And so we have an isolate collection. We'll grow everything on media, PDA, and then we'll sequence that. And we have different isolates that we've identified. There's also different species of Fusarium we've identified, but we don't normally do that. Like if there's a mapping population, we wouldn't do that from every sample. But we do that more broadly just to understand the diversity of the pathogen in the field.
Right, yeah. So me asking that was in order to prevent contamination and like narrowing down the specific pathogens that you're working with. Maybe I thought isolating them will like kind of like just narrow them down.
Yeah, so there's kind of multiple avenues of research. Like sometimes maybe we don't want to narrow it down. We want to understand in the field when you see an infected inflorescence, like what else is there? Maybe it is the co-infection that's actually causing the maximal expression. Like what is the difference between a 7 and a 9 in a rating? Like we're trying to understand if it is actually other organisms that are causing a difference in the phenotype. But then, the lab study, that's where we're like specifically, you're starting with the sterile media and inoculating it with a specific pathogen. And so that we do see totally different profiles there than we do in the field. And that is expected. But yes, you still see a lot of other stuff. But as I mentioned, the number of junk reads changes drastically. So if you have a really clean sample, you might have 92% of your reads mapped to Phaseolus vulgaris, and you have less reads to actually look at what is there. So I think we are doing a little bit of that. It's just I don't know if we might not only be interested in that alone.
Right, yeah. The last question was that in the greenhouse, what brings other pathogens? Is it maybe the source of water used to irrigate those seedlings?
If you have a perfectly clean greenhouse, let me know how you do it. But who knows? And you have different greenhouse pathogens than you have field pathogens, absolutely. So, we'll see thrips DNA or aphid, more greenhouse insect pets. You might see more in the greenhouse than you would in field samples. So it's just the different environments, but they're not closed greenhouses. They might get sprayed occasionally, but there still is quite a source of various things coming in and out of there.
Thank you.
Mm-hmm.
Yeah. Hi. I wanted to ask about something that you mentioned pretty early on in your talk. You said that if your variety had excessive THC, it would need to be destroyed. I wanted to know what a realistic risk that was. Like, if you or a colleague had any variety where, random variation led to excessive THC. You talked a little bit about how natural populations had different distributions of the trait, but I just wanted to know.
Yeah. So the interesting thing with the cannabinoid profile is type 3 plants, where the idea is that they have non-functional THCA synthase, they should be making no THCA. You still see THC production in those plants, and people attribute this to this leaky expression of CBDA synthase, sometimes accidentally making THCA in this like 1:25 ratio. So all CBD plants, if you grow them, you'll typically get some amount of THC in those plants. Now, if you knock out both of those synthases, you typically will get a large amount of CBG, and then you won't have that problem. And they make very low amounts of THC and CBD. But in the fiber and grain types, they normally have very, very, very low amounts of both CBD and THC, so they're much more likely to remain compliant. We talked about Wisconsin Crop Innovation Center and the talk previously, so there are people who are editing the pathway to knock out cannabinoid production completely, so those lines exist, that they're zero cannabinoid production lines, but they're edited, and there are naturally occurring CBG lines that are also safe to grow. So growers have some option, but I think one thing that is very clear now, if you're trying to maximize CBD production, you have to be very careful, because a lot of those cultivars will exceed that THC limit 5 weeks after flowering. So that's just a game that growers have to play with, testing at what time while they still remain compliant.
And so have you encountered that just directly? Have you known someone who's had to destroy their crop?
Many, many, many people. Yeah. So in the early years of production in Wisconsin, I was telling someone earlier, I think it was something like 14% of growers had to destroy their fields because they were non-compliant. And that's because they go to 0.36 or something. I mean, it's like a very, very small amount that they've accumulated over the legal threshold. But yeah, that's how it was then. I think everybody's kind of figured out how to deal with that in their own unique ways. Sometimes it's sampling time. Sometimes it's actually the cultivars themselves just don't produce as much, as high of cannabinoid content. If you were to change that level to where the legal limit was 1%, you would have much less concern with a lot of those CBD cultivars, because that limit is very rarely reached, even with the leaky synthase activity.
Thank you.
Mm-hmm.
Very nice talk. I really loved how you used the citizen scientists to isolate all those lines. That's really clever. Now, you said there are, in genetic diversity in the North American isolate that you just found. I'm just wondering if the source of this diversity is essentially a point of entry where there were multiple introductions, or you see any evidence of like diverging afterwards. After all, like 70 years is not that much, but something could have swept. So yeah.
Yeah, definitely. I mean, one of the populations that's very isolated is the New York feral population. So we kind of have this Indiana and these more Western, North Central, and the New York populations. And I do like to think about them in terms of where, there was the USDA breeding program and where material was sent. There's also feral populations in Canada. And I think these are probably different introgressions. So like when they became feralized, they were from different moments in time in the breeding program. There were a lot of Italian fiber types that were brought over that were then hybridized with Chinese fiber types. So you can see that in the literature. We haven't quite figured that out in the analysis, because there's so much hybridization as a dioecious outcrossing species. It's hard to capture that sometimes. But yes, I do think there were probably multiple escapes. But the material that they escaped from, I'm not sure how diverged those populations were. And yes, it's only been 70 years that they've been feralized. We do see, though, in our initial characterization, there is a very large inversion on chromosome 4 that segregates between the feral populations. So there are large-scale population dynamics that have evolved already in that time. Yes. And I believe we're almost at 4:20 right now, so we're very